A very good morning to madam. My name is Mugilan. My matrix number is 23DBT20F. Today I'm going to talk about the theory behind this experiment, which is running a protein gel. So, and about the materials for the simulations. Before that, we have to know what is SDS electrophoresis. So, protein gel electrophoresis is a laboratory technique that separates protein by mass. This technique is similar to running a agarose gel to separate and visualize different molecules of DNA. After electrophoresis, the otherwise invisible proteins are typically visualized by staining the gel with chromocy, which is generate the typical protein gel pattern of blue bands. However, when running a sample with many proteins such as an entire cellular extract, the visualization of individual proteins can be very tricky as hundreds of thousands of other proteins may uh, obscure the protein band you are interested in. Then we move to the materials that we need for the experiment. Uh, for the experiment, we need 4x SDS sample buffer solution, which is help to denature the solution. Then we need two control solutions, which is positive control and negative control. For the positive control, we use wild type cells culture. Then for the negative control, we use pancreas cell culture. Then we need the random samples for the other well of the gel. So which is edit 4 cell culture and edit 7 cell culture. Then you also need protein ladder solution. Then 1x running buffer for the gel electrophoresis process. Then we also need pipette, actually, especially for P20 pipette and also P200 pipette, and with its tip box. And we also need empty tubes to use to hold the solutions, which is wild type cell tube and pancreas tube, edit 4 tube and edit 7 tube. For the electrophoresis equipment, we need a polyacrylate gel and protein gel electrophoresis box and power supply leads. Then other equipments are microcentrifuge and biohazardous waste and heat block. Then for this experiment, I predict that the positive control would be the I predicted that the positive control solution is wild type cell culture and the negative is um, the pancreas cell culture. Then let's we move to the experiment of simulation. Before we going to see the simulation, we should read through the protocols of running a protein gel experiment. First and foremost, we have to collect cells, add sample buffer, then denature the proteins, centrifuge the samples, then load the gel and conduct protein electrophoresis. So let's start the simulation. So first and foremost, increase the heat block temperature to 95 degree Celsius. So add until 95 degree. Okay, we already achieved that 95 Celsius. Then close. Set a micropep to 30 microliters. So save the volume and attach a tip. Then take the wild type cell tube and the wild type cell culture and open the cell culture. and take that volume. Afterwards, close the wild type cell culture and put it back in the rack. Afterwards, open the wild type cell tube, then insert the solution inside. Okay, then close and put it back in the rack. Then we have to remove the tip. So for that, okay, then we have to repeat the procedure to the pancreas cell culture. The pancreas cell culture is over here and the pancreas tube over here. So we have to open the top and we have to put it the tip again. Then take the same amount of volume as before. Afterwards, open the pancreas tube and put the solution inside. Okay, then close the test tube, put the solution inside the rack, then close it. Then repeat the procedure to the edit for cell culture. Before that, we must eject the tip to avoid the contamination. Okay, then take the edit for cell culture, open it, and take the same amount. Then open the edit for tube and put the solution inside. Okay, then close the edit for tube and put it back in the rack. And lastly, we have to do the edits. We have to dispense edit 7 cell culture into its test tube. Okay, and open the tube. Okay, then close and put it in the rack. Then eject the tip. Then put it back. Okay, then put it back. Close that solution. Then let's move to the second procedure, which is add sample buffer. Let's move to the second procedure, which is add sample buffer. For that, we have to prepare a micropep to 10 microliters. For that, I took P20, then add 10 microliters. Okay, we already achieved the volume. Then attached the tip for it. Okay, then take the 4x SD sample, dispense the amount, and close the 4x SDS sample buffer. Then take the wild type cell tube, then dispense the SDS sample into the then close, then put it back in the rack. Then repeat the procedure with the following 3 solutions. Then take the pancreas tube, then dispense the SDS sample buffer. Then dispense the SDS sample buffer into the pancreas tube. Okay. Then eject the tip. So that close and put it back in the rack. Then let's move to the edit for tube. For that we have to take the new tip. Then dispense the 4x SDS sample buffer into the edit for tube. Okay, then eject the tip. Then put it back in the rack. Then take the edit 7 tube. Then eject a new tube. Then again dispense the 4x SDS sample inside the edit 7 test. Okay, then we already done the add sample buffer, so we have to put it back the micropipette, close, then close, put it back in the rack, then close, put it back in the rack. Okay, we already finished the second procedure. So let's we move to the denature protein procedure. For that we have to move all our samples to the heat block. Then we have to put timer for 10 minutes. Okay, then we have to start. Okay, afterwards we have to remove all the samples from the heat block. Okay, let's we move to the fourth procedure, which is centrifuge the samples. Move all our samples tubes to the microcentrifuge. So open the microcentrifuge and put the samples. Okay, check the balancing. So for that I put here for balancing. So check the balance. So centrifuge look balanced and ready. Then close. Then pulse. Then remove the tubes. Okay guys, then we move to the fifth procedure, which is load the gel. Place the polycarbonyl gel in the electrophoresis box. Afterwards, fill the gel cassette with 1x running buffer. Then remove the comb. Then we have to remove the comb. So we already remove the comb. Afterwards, we have to fill the outer gel electrophoresis tank with 1x running buffer. Then we have to take the buffer and put here. Then set a micropipette for 20 microliters. Okay, then save the volume. Then draw up the protein ladder. Okay, before that we have to attach a tip for it. Then open the protein ladder solution. Draw up the amount that needed. Okay, we already got the solution. And put it back. Then move the micropipette to the first lane of the protein gel. Okay. Dispense the protein ladder into the first wall. So how should you micropipe samples into the well? Quickly, so protein go down the well slowly to prevent spilling. Slowly but move the plunger. So this is the B. Okay. Then dispense the positive control into the second wall of the gel. So for that we take the wild type cell solution. So what we we choose the second. Okay, then done. Then we have to remove the tip. Then we have to draw the pancreas tube solution for dispense 20 microliters of negative control into the third well of the gel. After dispense the pancreas tube solution and put in the third well of the gel. So, okay, done. Afterwards, we have to dispense 20 microliters of edit for cells and want to dispense into the fourth well of the gel. So we have to take the edit cell tube. Then we have to eject the tip before it and take a new tip. Okay. Then dispense the amount. After that, put it back. Put that in the gel. In the fourth well of the gel. Okay, done. Then eject the tip. Then close the edit 4. Then let's move to the edit 7 tube, which is we have to dispense into the fifth well of the gel. So we have to put it the tip again. Okay, then take the amount that we need. Then put in the gel. Okay, done. So we already done the fifth procedure, which is the load the gel. So we have to put it back. Then the last procedure is conduct protein electrophoresis. For that we have to cover the electrophoresis box. Then connect the red wire into and the black cable into the black and put the right hole. Okay, then turn on the power supply. Then set the voltage to 150 voltage. Okay, then set a timer for 60 minutes. Then start the power supply. Then wait until the time has elapsed. Then how do you know if the gel has run for long enough? The die front is missing. The die front is at above the walls. The die from its bottom of the gel. The die front is across the ball. Correct. Yes, at the bottom. Okay, then turn off the power switch. Then we have to unplug the electrodes. Then we have to inspect our gel. Okay, then we already finished our running protein gel experiment. For the experiment, we get the actual results like this, and the ideal results would be like this. And the controls are positive control is wild type macrophage cells, and negative control is pancreas cells, and samples are edit4 cells and edit7 cells. And the Kumasi Saint gel is over CCR5. That's all from today. That's all from me. Thank you.